An acid is a molecule or ion capable of donating a hydrogen, or alternatively, capable of forming a covalent bond with an electron pair. The first category of acids is the proton donors or Bronsted acids. In the special case of aqueous solutions, proton donors form the hydronium ion H3O+ and are known as Arrhenius acids. Bronsted and Lowry generalized the Arrhenius theory to include non-aqueous solvents. A Bronsted or Arrhenius acid usually contains a hydrogen atom bonded to a chemical structure that is still energetically favorable after loss of H+. Aqueous Arrhenius acids have characteristic properties which provide a practical description of an acid. Acids form aqueous solutions with a sour taste, can turn blue litmus red, and react with bases and certain metals to form salts. The word acid is derived from the Latin acidus, acea meaning sour. An aqueous solution of an acid has a pH less than 7 and is colloquially also referred to as acid, while the strict definition refers only to the solute. A lower pH means a higher acidity, and thus a higher concentration of positive hydrogen ions in the solution. Chemicals or substances having the property of an acid are said to be acidic. Common aqueous acids include hydrochloric acid, acetic acid, sulfuric acid, and citric acid. As these examples show, acids can be solutions of pure substances, and can be derived from acids that are solids, liquids, or gases. Strong acids and some concentrated weak acids are corrosive, but there are exceptions such as carbarenes and boric acid. The second category of acids are Lewis acids, which form a covalent bond with an electron pair. An example is boron trifluoride, whose boron atom has a vacant orbital which can form a covalent bond by sharing a lone pair of electrons on an atom in a base. For example, a nitrogen atom in ammonia. Lewis considered this as a generalization of the Bronsted definition, so that an acid is a chemical species that accepts electron pairs either directly or by releasing protons into the solution, which then accept electron pairs. However, hydrogen chloride, acetic acid, and most other Bronsted-Lowry acids cannot form a covalent bond with an electron pair and therefore not Lewis acids. Conversely, many Lewis acids are not Arrhenius of Bronsted-Lowry acids. In modern terminology, an acid is implicitly a Bronsted acid and not a Lewis acid, since chemists almost always refer to a Lewis acid explicitly as a Lewis acid. Definitions and concepts. Modern definitions are concerned with the fundamental chemical reactions common to all acids. Most acids encountered in everyday life are aqueous solutions or can be dissolved in water, so the Arrhenius and Bronsted-Lowry definitions are the most relevant. The Bronsted-Lowry definition is the most widely used definition, unless otherwise specified. Acid-base reactions are assumed to involve the transfer of a proton from an acid to a base. Hydronium ions are acids according to all three definitions. Interestingly, although alcohols and amines can be Bronsted-Lowry acids, they can also function as Lewis bases due to the lone pairs of electrons on their oxygen and nitrogen atoms. Arrhenius acids. The Swedish chemist Svante Arrhenius attributed the properties of acidity to hydrogen ions of protons in 1884. An Arrhenius acid is a substance that, when added to water, increases the concentration of H+ ions in the water. Note that chemists often write H+ and refer to the hydrogen ion when describing acid-base reactions, but the free hydrogen nucleus, a proton, does not exist alone in water. It exists as the hydronium ion H3O+. Thus, an Arrhenius acid can also be described described as a substance that increases the concentration of hydronium ions when added to water. Examples include molecular substances such as hydrogen chloride and acetic acid. An Arrhenius base, on the other hand, is a substance which increases the concentration of hydroxide ions when dissolved in water. This decreases the concentration of hydronium because the ions react to form H2O molecules, H3O++ O minus H2O plus H2O. Due to this equilibrium, any increase in the concentration of hydronium is accompanied by a decrease in the concentration of hydroxide. Thus, an Arrhenius acid could also be said to be one that decreases hydroxide concentration, while an Arrhenius base increases it. In an acidic solution, the concentration of hydronium ions is greater than 10⁻⁷ moles per liter. Since pH is defined as the negative logarithm of the concentration of hydronium ions, acidic solutions thus have a pH of less than 7. Bronsted-Lowry acids. While the Arrhenius concept is useful for describing many reactions, it is also quite limited in its scope. In 1923, chemists Johannes Nicolaus Bronsted and Thomas Martin Lowry independently recognized that acid-base reactions involve the transfer of a proton. A Bronsted-Lowry acid is a species that donates a proton to a Bronsted-Lowry base. Bronsted-Lowry acid-base theory has several advantages over Arrhenius theory. Consider the following reactions of acetic acid, the organic acid that gives vinegar its characteristic taste. CH3COOH plus H2O, CH3COO minus plus H3O plus, CH3COOH plus NH3CH3COO minus plus NH plus. For both theories easily describe the first reaction. CH3COOH acts as an Arrhenius acid because it acts as a source of H3O+ when dissolved in water, and it acts as a Bronsted acid by donating a proton to water. In the second example, CH3COOH undergoes the same transformation, in this case donating a proton to ammonia, but does not relate to the Arrhenius definition of an acid because the reaction does not produce hydronium. Nevertheless, CH3COOH is both an Arrhenius and a Bronsted-Lowry acid. Bronsted-Lowry theory can be used to describe reactions of molecular compounds in non-aqueous solution of the gas phase. Hydrogen chloride and ammonia combine under several different conditions to form ammonium chloride. In aqueous solution, hydrogen chloride behaves as hydrochloric acid and exists as hydronium and chloride ions. The following reactions illustrate the limitations of Arrhenius's definition: H3O++ Cl- NH3Cl- NH4++ H2O. Hydrogen chloride plus NH3 ammonium chloride. Hydrogen chloride plus NH3 ammonium chloride. As with the acetic acid reactions, both definitions work for the first example, where water is the solvent and hydronium ion is formed by the hydrogen chloride solute. The next two reactions do not involve the formation of ions but are still proton transfer reactions. In the second reaction, hydrogen chloride and ammonia react to form solid ammonium chloride in a benzene solvent, and in the third, gaseous hydrogen chloride and NH3 combine to form the solid Lewis acids. A third, only marginally related concept was proposed in 1923 by Gilbert N. Lewis, which includes reactions with acid-base characteristics that do not involve a proton transfer. A Lewis acid is a species that accepts a pair of electrons from another species. In other words, it is an electron pair acceptor. Bronsted acid-base reactions are proton transfer reactions, while Lewis acid-base reactions are electron pair transfers. Many Lewis acids are not Bronsted-Lowry acids. Contrast how the following reactions are described in terms of acid-base chemistry. In the first reaction, a fluoride ion F- gives up an electron pair to boron trifluoride to form the product tetrafluoroborate. Fluoride loses a pair of valence electrons because the electrons shared in the BF bond are located in the region of space between the two atomic nuclei and therefore more distant from the fluoride nucleus than they are in the lone fluoride ion. BF3 is a Lewis acid because it accepts the electron pair from fluoride. This reaction cannot be described in terms of Bronsted theory because there is no proton transfer. The second reaction can be described using either theory. A proton is transferred from an unspecified Bronsted acid to ammonia, a Bronsted base. Alternatively, ammonia acts as a Lewis base and transfers a lone pair of electrons to form a bond with a hydrogen ion. The species that gains the electron pair is the Lewis acid. For example, the oxygen atom in H3O+ gains a pair of electrons when one of the HO bonds is broken and the electrons shared in the bond become localized on oxygen. Depending on the context, a Lewis acid may also be described as an oxidizer or an electrophile. Organic Bronsted acids, such as acetic, citric, or oxalic acid, are not Lewis acids. They dissociate in water to produce a Lewis acid, H+, but at the same time also yield an equal amount of a Lewis base. Few, if any, of the acids discussed in the following are Lewis acids. Nomenclature. In the classical naming system, acids are named according to their anions. That ionic suffix is dropped and replaced with a new suffix, according to the table below. For example, hydrogen chloride has chloride as its anion, so the -ide suffix makes it take the form hydrochloric acid. In the IUPAC naming system, aqueous is simply added to the name of the ionic compound. Thus, for hydrogen chloride, the IUPAC name would be aqueous hydrogen chloride. The prefix hydro is added only if the acid is made up of just hydrogen and one other element. Classical naming system. Acid strength. The strength of an acid refers to its ability, a tendency to lose a proton. A strong acid is one that completely dissociates in water. In other words, 1 mole of a strong acid hard dissolves in water, yielding 1 mole of H+ and 1 mole of the conjugate base, A-, and none of the protonated acid Ha. In contrast, a weak acid only partially dissociates, and at equilibrium both the acid and the conjugate base are in solution. Examples of strong acids are hydrochloric acid, hydroiodic acid, hydrobromic acid, perchloric acid, nitric acid and sulfuric acid. In water, each of these essentially ionizes 100%. The stronger an acid is, the more easily it loses a proton, H+. Two key factors that contribute to the ease of deprotonation of the polarity of the HA bond and the size of atom A, which determines the strength of the HA bond. Acid strengths are also often discussed in terms of the stability of the conjugate base. Stronger acids have a larger Ka and a more negative pKa than weaker acids. Sulfonic acids, which are organic oxyacids, are a class of strong acids. A common example is toluene sulfonic acid. Unlike sulfuric acid itself, sulfonic acids can be solids. In fact, polystyrene functionalized into polystyrene sulfonate is a solid strongly acidic plastic that is filterable. Superacids are acids stronger than 100% sulfuric acid. Examples of superacids are fluoroantimonic acid, magic acid and perchloric acid. Superacids can permanently protonate water to give ionic, crystalline hydronium salts. They can also quantitatively stabilize carbocations. While Ca measures the strength of an acid compound, the strength of an aqueous acid solution is measured by pH, which is an indication of the concentration of hydronium in the solution. The pH of a simple solution of an acid compound in water is determined by the dilution of the compound and the compound's Ca. Applications of acids. There are numerous uses for acids. Acids are often used to remove rust and other corrosion from metals in a process known as pickling. They may be used as an electrolyte in a wet cell battery, such as sulfuric acid in a car battery. Strong acids, sulfuric acid in particular, are widely used in mineral processing. For example, phosphate minerals react with sulfuric acid to produce phosphoric acid for the production of phosphate fertilizers, and zinc is produced by dissolving zinc oxide into sulfuric acid, purifying the solution and electrowinning. In the chemical industry, acids react in neutralization reactions to produce salts. For example, nitric acid reacts with ammonia to produce ammonium nitrate, a fertilizer. Additionally, carboxylic acids can be esterified with alcohols to produce esters. Acids are used as additives to drinks and foods, as they alter the taste and serve as preservatives. Phosphoric acid, for example, is a component of cola drinks. Acetic acid is used in day-to-day life as vinegar. Carbonic acid is an important part of some cola drinks and soda. Citric acid is used as a preservative in sauces and pickles. Tartaric acid is an important component of some commonly used foods like unripened mangoes and tamarind. Natural fruits and vegetables also contain acids. Citric acid is present in oranges, lemon and other citrus fruits. Oxalic acid is present in tomatoes, spinach, and especially in carambola and rhubarb. Rhubarb leaves and unripe carambolas are toxic because of high concentrations of oxalic acid. Ascorbic acid is an essential vitamin for the human body and is present in such foods as amla, lemon, citrus fruits, and guava. Certain acids are used as drugs. Acetylsalicylic acid is used as a painkiller and for bringing down fevers. Acids play important roles in the human body. The hydrochloric acid present in the stomach aids in digestion by breaking down large and complex food molecules. Amino acids are required for synthesis of proteins required for growth and repair of body tissues. Fatty acids are also required for growth and repair of body tissues. Nucleic acids are important for the manufacturing of DNA and RNA and transmitting of traits to offspring through genes. Carbonic acid is important for maintenance of pH equilibrium in the body. Acid catalysis acids are used as catalysts in industrial and organic chemistry. For example, sulfuric acid is used in very large quantities in the alkylation process to produce gasoline. Strong acids, such as sulfuric, phosphoric and hydrochloric acids also affect dehydration and condensation reactions. In biochemistry, many enzymes employ acid catalysis. Biological occurrence. Many biologically important molecules are acids. Nucleic acids, which contain acidic phosphate groups, include DNA and RNA. Nucleic acids contain the genetic code that determines many of an organism's characteristics, and is passed from parents to offspring. DNA contains the chemical blueprint for the synthesis of proteins which are made up of amino acid subunits. Cell membranes contain fatty acid esters such as phospholipids. An alpha amino acid has a central carbon which is covalently bonded to a carboxyl group, an amino group, a hydrogen atom and a variable group. The variable group, also called the R group side chain, determines the identity and many of the properties of a specific amino acid. In glycine, the simplest amino acid, the R group is a hydrogen atom. but in all other amino acids it contains one or more carbon atoms bonded to hydrogens, and may contain other elements such as sulfur, oxygen and nitrogen. With the exception of glycine, naturally occurring amino acids like aryl almost invariably occur in the L configuration. Peptidoglycan, found in some bacterial cell walls, contains some D amino acids. At physiological pH, typically around 7, free amino acids exist in a charged form, where the acidic carboxyl group loses a proton and the basic amine group gains a proton. The entire molecule has a net neutral charge and is Witterian, with the exception of amino acids with basic or acidic side chains. Aspartic acid, for example, possesses one protonated amine and two deprotonated carboxyl groups, for a net charge of -1 at physiological pH. Fatty acids and fatty acid derivatives are another group of carboxylic acids that play a significant role in biology. These contain long hydrocarbon chains and a carboxylic acid group on one end. The cell membrane of nearly all organisms is primarily made up of a phospholipid bilayer, a micellar of hydrophobic fatty acid esters with polar, hydrophilic phosphate head groups. Membranes contain additional components, some of which can participate in acid-base reactions, in humans and many other animals. Hydrochloric acid is a part of the gastric acid secreted within the stomach to help hydrolyze proteins and polysaccharides, as well as converting the inactive proenzyme pepsinogen into the enzyme pepsin. Some organisms produce acids for defense. For example, ants produce formic acid. Acid-base equilibrium plays a critical role in regulating mammalian breathing. Oxygen gas drives cellular respiration, the process by which animals release the chemical potential energy stored in food. Producing carbon dioxide is a byproduct. Oxygen and carbon dioxide are exchanged in the lungs, and the body responds to changing energy demands by adjusting the rate of ventilation. For example, during periods of exertion the body rapidly breaks down stored carbohydrates and fat, releasing CO2 into the bloodstream. In aqueous solutions such as blood, CO2 exists in equilibrium with carbonic acid and bicarbonate ion. CO2 plus H2O H2 CO3 H plus HCO3 minus. It is the decrease in pH that signals the brain to breathe faster and deeper, expelling the excess CO2 and resupplying the cells with O2. Cell membranes are generally impermeable to charged large, polar molecules because of the lipophilic fatty acyl chains comprising their interior. Many biologically important molecules, including a number of pharmaceutical agents, are organic weak acids which can cross the membrane in the protonated, uncharged form but not in the charged form. For this reason the activity of many drugs can be enhanced or inhibited by the use of antacids or acidic foods. The charged form, however, is often more soluble in blood and cytosol, both aqueous environments. When the extracellular environment is more acidic than the neutral pH within the cell, certain acids will exist in the neutral form and will be membrane-soluble, allowing them to cross the phospholipid bilayer. Acids that lose a proton at the intracellular pH will exist in the soluble, charged form and thus able to diffuse through the cytosol to the target. Ibuprofen, aspirin and penicillin are examples of drugs that are weak acids. Common acids. Mineral acids, hydrogen halides and the solutions: hydrofluoric acid, hydrochloric acid, hydrobromic acid, hydroiodic acid. Halogen oxoacids, hypochlorous acid, chlorous acid, chloric acid, perchloric acid, and corresponding compounds for bromine and iodine. Sulfuric acid, fluorosulfuric acid, nitric acid, phosphoric acid, fluoroantimonic acid, fluoroboric acid, hexafluorophosphoric acid, chromic acid, boric acid, Sulfonic acids. A sulfonic acid has the general formula Rs2OH, where R is an organic radical. Methane sulfonic acid, ethane sulfonic acid, benzene sulfonic acid, betoluene sulfonic acid, trifluoromethane sulfonic acid, polystyrene sulfonic acid SO3HN. Carboxylic acids. A carboxylic acid has the general formula RCOH, where N is an organic radical. The carboxyl group COH contains a carbonyl group C equals O and a hydroxyl group OH. Acetic acid, citric acid, formic acid, gluconic acid OC2, 4Cu, lactic acid, oxalic acid, tartaric acid, halogenated carboxylic acids. Halogenation at alpha position increases acid strength, so that the following acids are all stronger than acetic acid: fluoroacetic acid, trifluoroacetic acid, chloroacetic acid, dichloroacetic acid, trichloroacetic acid, vinylogous carboxylic acids. Normal carboxylic acids of the direct union of a carbonyl group and a hydroxyl group. In vinylogous carboxylic acids, a carbon-carbon double bond separates the carbonyl and hydroxyl groups. Ascorbic acid, nucleic acids, deoxyribonucleic acid, ribonucleic acid,